Namaste students, for sure Vidya. I'm JVN Dr. Rajendra, Assistant Professor to JVWU, University Jaipur. In today's session, we are going to discuss about the linkage, genetic linkage and crossing over. Okay. So, first of all, is independent assortment always the case? That means the independent assortment states that during gamete formation, the two alleles for one gene segregates or assorts. Okay, that means independently of the alleles of the other gene. But if two are found on the same chromosome, then they are not assorted independently. That means they do not follow the Mendelian inheritance pattern. Right here. Okay. That means the genes that are inherited together are said to be the linked. Okay. Okay. So here that means independent assortment states that during gamete formation, two alleles for one gene segregate or assort independently of the allele for other genes. But if two genes are on the same chromosome, they will not assort independently. So this you have to mark, okay? And do not follow Mendelian inheritance principles or a pattern here. That means the genes are inherited together and are said to be linked. The genes located on different chromosomes are not linked. This allows independent assortment in dihybrid cross. Okay? The tree shows that the classical 9 is to phase to 1 inheritance pattern. Okay? That means the genes that are located very close together on the same chromosome may show complete linkage. They may not so close each other that they cannot be separated by recombination during meiosis. Okay. So here the gene located for far apart on the same chromosome typically shows incomplete partial linkage because they are easily separated by the recombination. Okay. Right? So here a linked gene, a unlinked gene. Yeah. Here, let me share. So here you can see the A, B, and C are the linked genes. Okay. So let me share the screen. You can see. So here you can see in this screen the A, B, C. That means the linked gene, and here we have a pair of homologous chromosomes. Okay. So these pairs of homologous so here you can see the A, B, and C. These three are linked genes on the same chromosomes. But here the loci that is A and B are not linked. B and D. It is on different chromosomes. So here these are not the linked one, right? This is unlinked gene. A and D, whereas A, B, C are linked chains. So here we call these as linked chain, and whereas A and D are the unlinked chain, that means they are not on the same chromosomes. Okay. So here you can see the genetic linkage, or the linkage is the genetic linkage is the tendency of an allele that are allowed close together on the chromosomes to be inherited together during the meiosis. Okay, that means the gene whose loci are nearer to each other are less likely to be separated on the different chromatid during chromosomal crossing over, or they are said to be genetically linked. In other words, the nearer two genes are on the chromosomes, and the lower of the chance of swam or occurring between them. They are more likely to be inherited together here. Okay? So the discovery first, when we say the first, the William Batson and Punnett completed a study in 1908 that determines the movement of an allele found on the same chromosome. That means the study used on the sweet pea, particularly the flower color and the pollen shape. They followed the inheritance pattern, right? So these patterns forming for flower color or the purple or dominant over the red, and the pollen shades long is dominant over the round. Okay. So a cross was formed using a true breeding purple or a long or a round. That means the F1 generation was 100% purple long, right? So here the crossing over of the two individuals from the F1 generation resulted in the F2 generation with 4 different phenotypes. Okay, that means the purple, long, red, or red brown. Okay, right? So the alleles that created this combination did not follow 9:3:3:1 pattern, but supported this idea the alleles do not assort independently and must be linked. Okay, so here you have to see the crossing over which was on the two individual forms, the F1 generation resulted in that of the F2 generation with 4 different phenotypes. So here are the 4 different phenotypes, that means the purple long and the purple brown, or the red long and the red brown. That means these alleles that was created, this combination do not follow this 9:9:3:1 pattern. So it is to about support the idea of that the alleles did not independently assort and therefore must be linked. Okay, so when we see the linkage part of the form of crossing over parts, okay, wait, here. So here you can see the phenotypic appearance of this was the purple colored flower with long pollen and the red flower with round pollens. Okay, so here these long flower when crossed with these red flowers. Okay, right? So here the structure here was red and it was purple. Okay, this is the purple one and this was the red one. So here in this, the combination was the purple long, that is capital P, small p. Okay. So here you can see the combination was formed with capital P, and long. Okay, which was crossed with, and forming the cross with small p, and forming small p, smaller, smaller. Okay. That means the next combination developed was capital P, small p, and capital L, small l. Right? So here this combination occurs during after the in the meiosis. Okay? So here the F1 offsprings, here the F1 offsprings, okay? The F1 offspring here formation of in the period generation. Okay? So which allows in the F2 offspring you can see the formation of the purple flower, which was again crossed between purple long into purple long, and which gave rise to 15 different combinations. Okay. So the chromosomes are for the organizing here. These chromosomes are for organizing the gene for the safe coordination of the transmission from cell to the cell. Okay. That means here when we see the type of linkage, depending upon the presence or the absence of the new combinations or the new parental combination, or the linkage can be of two types. That means the complete linkage if two or more characters are inherited together and consistently appears in two or more generation in the original or parental combinations. Okay, that means it is called the complete linkage. These genes do not produce non-parental combinations, and the genes showing the complete linkage are closely located in the same chromosomes. Okay, this much is understood. So here next is incomplete linkage, when the linkage is exhibited by the those genes which produce some percentage of the non-parental combination. That means such genes are located distantly on the chromosomes. It is due to the accidental or occasional breakage of the chromosome segment during the crossing over. So, okay. So what are the significance of the linkage? Here the linkage do not permit the breeder to bring the desirable character in one variety. For this reason, the plants and the animal breeders find it difficult to combine various characters. Okay? So here the linkage reduces the chance of the recombinations of the genes and thus help in hold the parental characters together. So it is thus help in the organisms to maintain its parental or racial or other characters. So the linked genes are not inherited together every time. That means the chromosomes exchange from homologous genes during meiosis. Okay. So the crossing over and the inheritance of the linked gene, which do not always stay linked. These linked groups get suppressed by crossing over during the prophase 1 of the meiosis. Okay. That means no crossing over of the daughter cells are identical to that of the parent part. All the crossing over occurs cause genetic variations in the daughter cells are not identical to the parent one. Okay. So here when the crossing over occurs, the genes that are previously linked become unlinked, creating four different types of chromosomes, and these portions are not equally because the crossing over do not occur in every cells. That happens during meiosis. Okay. So here in the crossing over, you can see there is a random exchange of the DNA take place. Okay. In between the two non-sister chromatids of the homologous chromosomes. Okay. Which results here, which results in the here which results in the formation. Let's see. So here you can see the crossing over. It's a random exchange of DNA between the two sister chromatids. Okay. And two sister chromatids are randomly exchanging between the, okay, and the result is the combination of the genetic material. So here you can see the genetic combination, the genetic factor. That means here you can see the different combination has been formed. So here the pairing has been taken place, and in this point, the two between the two non-sister chromatids. Okay. These are the non-sister chromatids of the homologous chromosomes, which results in the combination of the new genetic material, right? That means there is the 50% combination of the material, right? Okay. So here the recombination is dependent on the distance between the linked gene. So what is the distance between the two genes which have been linked here? That also determines the crossing over, right? So what are the factors which affects this crossing over? That means the sex. That means their tendency of reduction of the chromosome over in male mammalians. Mutations reducing the crossing over. Temperature, that means high and low temperature variations, increasing the percentage of the chromosomes over a certain period of the chromosomes. X-ray effects, the X-ray irradiance or the increasing crossing over near the centromere age. Older age increases the rate of the crossing over. So what are the significance you can see over the crossing over? Produces new combination of traits. Though there is a like crossing over, the segments of the homologous chromosomes are interchanged, which forms in the original or the new genetic variation. So the crossing over may play an important role here in the breeding to improve the variety in plants, in animals also. The crossing over may help in breeding. Okay. So there. So different linkage is the tendency of a gene on a chromosome to remain in together or pass it to the next generation. That means it brings about the parental type. Strength of the linkage between the two genes increase and they are closely placed on a chromosome. It helps in mainly in the newly or new improved variety, right? So here the crossing over, it is exchange of the gene or the chromosome part to break the establishment of the linkage and form of the new linkage. It produces the recombination or the frequency of the crossing over between the two genes, or it is the source of the variations for the newly produced variety. So the parental genetic materials are the gametes that maintains the original linkage of the gene, right? Thank you. This session is powered by 2.0 VIDYAPEETH, Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session.